Now we want to apply these ideas about redox reactions to electrochemical cells. So we said that redox reactions tend to be really spontaneous. There's a large energetic driving force for this idea of transferring electrons. And so moving electrons can be useful, right? On some regards, it can be things like, there's a lot of energy if you just burn, say, propane, or you get pretty colors in your fireworks. But these sorts of redox reactions are really aggressive. And so if you could harness that flow of electrons, it would be really, really useful. So we do that harnessing using what's called an electrochemical cell. These are also called galvanic or voltaic cells. So electrochemical is kind of the descriptive name. Galvan and Volta were scientists who worked on this, so kind of naming it after them, or kind of the more electrochemical meaning kind of generally what's going on. It's chemistry driving electrons. And so what we're really looking at here is the idea of separating out the half reactions that are involved in the reaction. So we could have, say, our oxidation half would be like zinc here, and our copper would be our reduction half. And instead of those occurring in the same container, they are in separate beakers. And what you can basically do is funnel the electron flow through something useful, like turning on a light. And these reactions that we're doing via cells could still happen in one pot. So for example, if you mix zinc and copper, you put your zinc metal in your copper solution, it very quickly you see reaction that you start to see copper solid basically attaching onto the surface. So something like this here, that's all of this basically black solid that you're seeing right here. And you can also note that the blue solution becomes much more colorless because these copper cations that were in solution are attaching down onto the surface, right? Because they're undergoing that reduction process. And so this is a really aggressive reaction. The idea of a cell is take the same chemistry, but instead of just letting the flow of electrons happen, just right between the atoms, make the electrons pass through a wire, in which case you can harness those electrons. So here is our schematic of a cell. The parts of the cell is you're going to have your electrodes. This is going to be some sort of conductive material, that means metal, that goes between, that basically is how electrons flow out of the half reaction. So basically, this is usually a metal that's participating in the reaction. It doesn't have to be, but if there is a metal participating in half reaction, say for example, the zinc or copper, you just use a piece of that. Then you have the electrolyte. That's the solution that your electrode is sitting in. There's always going to be some sort of aqueous reactant or product that's involved. And so you want to be sure that you have that dissolved. You do want to make sure that there are ions in the solution because, again, you're looking for flow of electron. So there needs to be something, you need to have an electrolyte solution that can carry charge. You do also want to note that when we're looking at, say, something like, copper 2+ is, say, a reactant here. In order to get that dissolved in solution, you do need a counter ion. And that counter ion is just going to be something that's non-reactive. So something like sodium, like what we saw with like acid-base discussion, just doesn't do a lot of chemistry. Sulfate would then be our choice for non-reactive as far as redox chemistry for an anion. There are other choices as well, but those two always work. All right, so whenever you have a cell, one of your half reactions is going to be the oxidation reaction, and one is going to be the oxidation reaction. You need to have both of them, and we'll learn eventually how to predict. For now, we're just going to know that copper is going to reduce, zinc is going to oxidize. So the zinc reaction here in this case is going to be my oxidation. We call the half reaction, the electrode where oxidation occurs, is called the anode. Okay, so one of the ways you can remember that little mnemonic is an-ox. So 2 words, an-ox, anode, oxidation. And the electrons flow away from this anode. So see that right here. That is because the oxidation is lost. Your reactant is zinc metal. Those atoms of zinc metal are going to react and lose electrons. So they're going to flow out into the wire up from your electrode. As your reaction happens, you're going to lose reactant. Reactant is this piece of metal, meaning that if your electrode is participating in the anode reaction, then you're going to lose mass. So we would expect this piece of zinc to get smaller as you go along, because it's going to lose atoms, it's going to lose mass. Reduction happens at the cathode. Red cat, we can remember, so it's anox for the anode, red cat for the cathode. And here we can see the electrons are flowing in, consistent with the idea that reduction is the gain of electrons. So there are copper cations floating around in solution that will combine with these incoming electrons to produce copper solid. As a result, this electrode gets bigger, right? It starts to grow, it gets bigger, more and more atoms would be attaching on because your product is copper. And as a result, your electrolyte solution is going to decrease in concentration. You're going to lose copper cations floating around in solution. So if we, are kind of looking at this, we think about this overall process, our cell, chemistry is going to happen. And one of the things that we can see is going to happen is we're going to start to make cations and consume cations. So in a single kind of beaker reaction, that would balance it out. But this is going to be a problem in a cell because you would just have, say, additional cations in this beaker without any anions to counter them. And here, there would be additional anions in this beaker, and that's not going to work. This would cause a charge imbalance. Basically, once you have extra cations or extra anions, basically the system is just going to stop. You need to close the circuit, basically, to account for the cation imbalance. And that's where your salt bridge comes in. There is basically some sort of tube, porous media, something that is basically soaked through with a solution of salt. So we call it a salt bridge. It's the salt, you just need ions, electrolytes, and bridge, it just connects the two. But importantly, the only thing that flows through it is ions, not electrons. And so the ions can flow wherever is necessary to basically counter that charge. You just have all these extra, this is kind of just a reservoir of ions, and they can go either which way to basically just balance out the charge, keep everything at the concentration that basically one cation per anion, kind of accounting for those sorts of things, making its balance. But you always need a salt bridge for any electrochemical cell. So this is kind of an overall diagram, a fancier kind of depiction than my MS Paint one of the copper zinc reaction. The zinc is, electrons leave the zinc, go into the copper. You'll note it's a different depiction between what's on the left and what's on the right. There's no kind of set guidance that the anode has to be on one side or the other. That's not the case. What is going to be the case in this reaction is that when you're pairing zinc and copper, zinc is always the anode. And so the electrons always have to flow from anode to cathode, from zinc to copper. We can denote electrochemical cells using what's called cell notation, where basically we have the anode material, so the actual electrode, whatever's in the electrolyte, then the cathode electrolyte, then the cathode. This is literally, you'll note this is the oxidation over here. So this is where the electrons are coming from. The cathode is the reduction. That's where the electrons are going to. So we have this this is literally being written in the order of how the electrons are moving. You see there's double lines in here that can also be replaced by explicitly saying what the salt bridge is. So you can put double lines that just implies there is a salt bridge because everyone would know there has to be a salt bridge for your cell. You can also specify what it is. So, you could say it's sodium sulfate or whatever you're using. Sodium sulfate would definitely be the most common. But anode's always first because that's where electrons start or flow from. Cathode's always second. That's where electrons go. So we can take a look at our cell. We said that zinc was going to be our anode, so it goes first, and we put the actual metal first. Then we have the zinc 2+. You'll note here that this is a little bit new in notation. We're not only saying that it's aqueous, we're also specifying what the concentration is. We're going to see later on that concentrations are going to be really important when we're talking about cells. And so as a result, we do get that, we do include that a lot of the times for the specifications. Copper 2+, then with the 1 molar, and then the solid electrode for your cathode as well goes last. And one of the ways you can also understand it is basically this is reactant to product, reactant to product. This is the overall flow of electrons. You can also explicitly include the salt bridge. Sodium sulfate is the most common, but the important thing is that all of this picture is going to be depicted in this one little line. So overall, we have oxidation at the anode. That's where our reactions start. Electrons flow away. These electrodes lose mass. The electrons flow over to the cathode. They don't have to flow through a useful thing. You wouldn't go to the trouble generally of doing the chemistry this way, but the electrons are always going to flow into the cathode. The electrons always flow from anode to cathode. Reduction happens there, and we can see that the mass would be increasing. And so when we say that this mass changes, it really is, when you're losing all these atoms of, say, zinc here, your electrode is going to start to look like this. You would, after the reaction's over, you would have less reactant. Zinc electrode is part of your reactant. Copper is a product. That bad boy gets bigger. The electrons are the atoms are coming out of solution. There's fewer dissolved ions. They're going on to the surface of the electrode. All these depictions we've looked at have seemed like kind of fancy electrodes and all this kind of stuff, but when you're doing this stuff in lab, this doesn't need to be fancy. You can see these are just kind of folded pieces of like copper foil. Zinc is tougher, so it's going to stand up straight. These are just alligator clips. This is literally a piece of paper for a salt bridge, and that's really all it needs to be. Some like paper towels work really well, just because basically you can think about it, they soak up the salt water basically that you have. The ions can flow, but there's no electrons flowing through there. It's not like you have charge flowing. It's not a wire or anything like that. You just have kind of those ions moving through, and it really doesn't. We have all these kind of fancy images, but really these are just beakers, right? These aren't even fancy containers. For this crumpled up piece of copper. This doesn't have to be fancy. The chemistry still is going to happen. Again, these things aren't picky. This is spontaneous as it gets. Participation 4.3, question 3. Complete each of the following statements. 1, blank occurs at the anode. 2, an galvanic cell of half reaction Mg solid becomes Mg2 plus aqueous, and 2 electrons would occur at the blank. 3, while using a galvanic cell, the mass of the anode blank. 4, a galvanic cell requires a blank bridge in order to maintain a neutral charge in each half reaction. This is the third question on participation 4.3, due Friday, April 3rd at 11:59 p.m. over on Blackboard. Link to the assignment is right below the document where these videos are posted. Again, complete each of the following statements. Blank occurs at the anode. In a galvanic cell, the half reaction magnesium becomes magnesium cations and 2 electrons would occur at the blank. While using a galvanic cell, the mass of the anode blank, and a galvanic cell requires a blank bridge in order to maintain a neutral charge in each half reaction. So we can set up these cells. We know the parts, we know the language, but the whole point of this was to use them to get these useful electrons. So if we're going to use it, how quickly are these electrons going to flow? Why do they flow from zinc to copper, right? I just said that was a thing. Why? How can we predict that? How bright can we get a bulb to be? How can I make it better? How can I optimize? So we're going to spend a lot of time talking about how to use and better kind of utilize and kind of treat the cells that we can kind of construct here now from our half reactions. That's it. 3 questions on participation 4.3. You want to find all 3 of those questions, get them answered, and get those answers submitted over on Blackboard by Friday evening, Friday night. And then this weekend, you just got the homework due Saturday and the post lab due Sunday. Hopefully, what we did in the redox titration lab now makes a bit more sense. If you have any questions about anything, let me know. Otherwise, enjoy your weekend. Oh, the answers to the participation will go live Friday night. Encourage you to check those out. Otherwise, enjoy your weekend. Bye-bye.